everybody. Today I'm going to talk to you about MRSA, which is known as methicillin-resistant Staphylococcus aureus, and this disease has been increasing in inpatient and outpatient facilities within the last few years. It is spread through direct contact, which usually ends up being the healthcare or provider's hands, and it can also spread through people that are carrying the disease but not infected by the disease. So in order to start testing for MRSA, a swab of the anterior part of the nose is needed. This is just the very surface of the nose. It doesn't take too much. And then after that, once the swab is completed, a broth enrichment step is needed. So the swab is put into a broth and incubated overnight to see the growth. After this, then we have to plate the bacteria. So 200 milliliters of bacteria is pipetted onto a plate, which you can see in the top picture. And then this is also incubated overnight. There is usually antibiotics on this plate. And then wherever the bacteria does not grow is known as the zone of inhibition. So the bottom picture shows that. This tells you what antibiotics the bacteria is resistant to or not. The next step in detecting MRSA is the polymerase chain reaction, which is also known as PCR. The goal of PCR is to target the genes MEK-A and FEM-B and make many, many copies of this. So then colonies that grew on the plate are chosen to be plucked from the plate and put into 50 microliters of water. The tubes are run through the thermocycler, which you can see on the picture on this slide, at different temperatures, which then does different things to the DNA. After 30 cycles, these fragments of DNA are visualized using gel electrophoresis. The goal of gel electrophoresis is to separate nucleotides based on their fragment size. So once a gel recipe is made, it is poured into the mold of the electrophoresis and left to solidify at room temperature. And then the gel is set into the electrophoresis tank, and then 10 microliters of PCR product is pipetted into each of the slots, as you can see on the picture right here. Bacteria is put into those slots, and then in one slot a DNA ladder is put, and the DNA ladder helps with measuring the base pairs of FEM-B and MEK-A. And then the tank is filled with some buffer and the current is sent through it, and then after 45 minutes the gels can be transferred off of the mold and placed under a UV light. The UV light helps see how far the bands of DNA traveled down the gel. So the smaller the fragments, the farther the fragments will travel. So in this case, MEK-A is 310 base pairs and FEM-B is 651 base pairs. So MEK-A travels farther down the gel than FEM-B will. So then you can look under the UV light, and if FEM-B and MEK-A are present, then that slot containing that bacteria is positive for MRSA. So, is universal screening for MRSA worth it? Although this may be the best way to prevent the spread of MRSA, screening every single patient for MRSA is way too expensive for many and most hospitals. James A. McKinnell is an infectious disease specialist. He works for the LA Biomedical Research Institute, and he was the lead researcher for a MRSA project. The project looked at and included hospitals in Orange County, California, and they looked at the cost of testing all patients for MRSA. The swabbing was done the most simplest way, which is just the nares like I talked about earlier, and it costed hospitals on average $103,000 per 10,000 admissions. A quote from McKinnell says, "Although more extensive MRSA testing and isolation could prevent hospital-acquired MRSA infections, we found that the cost of such a program far exceeds any savings to the hospital. Our results are surprising because we know that preventing MRSA infections is better for the healthcare system as a whole, but the rewards of this effort do not seem to come back to the hospital in a meaningful way." Overall, there are better and more cost-effective ways that we can prevent MRSA, such as cleaning our hands with soap and water, and this can be healthcare workers and just people in the public as well. Cleaning hospital rooms and medical equipment, covering wounds, and so that bacteria is not transferred in and out of that wound, and only test for high-risk patients for MRSA. So high-risk patients include catheters and dialysis patients. So these ways are much better than just testing every single patient that comes into the hospital. It's more cost-effective, and there are other ways that MRSA can be prevented.